In this video, we'll focus the differences between SEM and TEM. If you see the scanning electron microscope from up to the source, SEM and TEM both are same. Source, we use a filament and acceleration anode. Both are common in SEM and TEM. But if going to the condenser lenses and object lenses, this is the only difference. Condenser lenses is the same position. If you see condenser lens, objective lens, and the focus onto that sample. By going to the TEM, the condenser lenses, and in between after condenser lenses there is a sample. After that objective lenses and focusing on projector in the TEM. Here it is a sample position is differs in the SEM and TEM. In SEM, sample is placed after all the lenses. But in case of TEM, sample is placed in between the condenser lens and object lens. After that, emitted light ray goes and form that image on the phosphor screen. But here it is in a scanning electron microscope. The sample is the ray beam, the means emitted ray beam from that filament is interact with the sample, and it generates that X-rays, reflected and refracted X-rays sample and secondary electrons. In SEM, we see that the beam interact with the surface of the sample, and reflected ones we study the profile of surface profile of the sample. But in case of the TEM, we'll see that the light rays passing through the sample and it form the image upon that phosphor screen. That image profile we'll see. This image is a three-dimensional profile. Here it is in SEM, the image profile because of secondary electrons in CRT and fluorescent screen, we'll see that. Fluorescent screen will give that image on that screen, the LED display. We can see that only two-dimensional image, and also that's a surface only, but not inside that material. But in case of TEM, the light ray passing through the sample, so we can see the three-dimensional image of the sample, and also we can see the each and every grain size inside the sample, and grain morphology means grain structures, whether it is a wire-like structure or balls-like structure, different structures we can see in case of transmission electron microscope. But in case of SEM, only reflected rays we study, it will give that only surface profile. And also because of that electron beam interacting with any material, then automatically X-rays will be produced. This is the X-rays which are produced. Okay, these are the major differences between SEM and TEM. You have to remember the sample is placed in between in TEM, but in SEM outside of